Hello, my name is Connie Liu. I'm an undergraduate at MIT, and today we're going to be talking about a concept question for cytoplasm and cytoskeletons. For today, we're going to be mostly focusing on cytoskeletons because they're the more interesting part of it. So we're going to be doing a basic comparison of all of the polymers involved in the cytoskeleton structure. So first, let's give another look to microtubules. So if you remember, these are the largest of the filaments in the cytoskeleton. And they kind of look like hollow tubes, which consist of 13 columns of tubulin molecules. So essentially, they kind of look like a spiral in that there's just columns of these tubulin dimers that form a tube for the structure to be able to hold up the whole cell. These microtubules are mainly involved in maintaining the cell shape. As you can tell, they're very, very strong, and they help out with cell motility as well. So microtubules, because they're so strong and so able to hold up their own weight, are also very involved in cell motility. So cilia and flagella are also made of the same things as microtubules. They're able to withstand the more usually harsher conditions outside of the cell and be able to sit outside the cell and allow for cell motility. So a lot of prokaryotes or bacteria sometimes have either cilia, which are smaller, shorter little appendages to the cell that allow it to kind of crawl or like tickle along, whereas flagella wave along and then kind of take that motion. So this is what you see in, say, sperm. An example where you can find cilia is bacteria. So yeah, those are very commonly used in those types of uses as well. But they can also help out with moving around organelles just inside the cell. So what microtubules can actually do is they can take themselves apart and move themselves to another part of the cell in order to help different organelles move around the cell, which is pretty cool, how they can disassemble and assemble by themselves. Now we're going to take another look at microfilaments. So these are the smallest, and they're made of actin, which is found in your muscles. So these help a lot with muscle contraction, or just like cell contraction and cell motility. They're also really involved in cell division. They help form the cleavage furrow that allow for two cells to be created out of one and them to pinch off from each other. So they are also very important. And they're just made out of two intertwined strands of actin. So that kind of looks like DNA, but they're just two strands that are twisted together to create microfilaments. And then we're going to look at intermediate filaments. And intermediate filaments are a bit more fibrous, and they're made of proteins that are super coiled into thicker cables. So we would have a cable like this, and then they would just be coiled really, really tightly. So if you think of a rope that's coiled really tightly and like kind of look like that, and then it's just like a lot of filaments twisted all together. And these are medium in size compared to microtubules and microfilaments. And for the most part, they are also involved in maintaining cell shape, just like the other two, but they also anchor the nucleus and other organelles within the cell, which is very important. And then they create the nuclear lamina. So the nuclear lamina is the kind of nuclear envelope that surrounds the nucleus inside the cell. So it plays a large role in that. And then a lot of the protein that intermediate filaments are made out of is keratin, which is very, very important for structure. It's the only structural part that still exists after the cell dies. So even dead cells have structure because of the keratin or the intermediate filaments found within them.